we're going to start with one of our favorites, which is, of course, carbohydrates. Okay. So, carbohydrates, the basic things, that's what most of our food grains are. Corn, rice, wheat. It's mostly carbohydrates, starch. We'll talk about the chemistry of that. And we use these to make bread and pasta. And another major carbohydrate is potatoes, which is just another form of starch. And these are all good ways of getting energy and molecules we need to build our bodies. We also get a little bit of it, very few people in this room, but a lot of people get their major carbohydrate in this form. Kind of little bit of food color, otherwise just straight sugar. Okay. And in terms of kind of nutritional problems, this is of course a major problem in the United States and the world. And let me at this point put in what I should have started with last time. The certain disclaimer is that my degree is a PhD and not an MD, okay? And so I won't be able to help myself once in a while giving some nutritional advice, but be aware that I'm not really a certified nutritionist. I have read a lot, I know the biochemistry, but please don't take what I say as a medical directive. Okay, but the problem here is not that sugars are bad for us, but that we have way too much of it in the diet, and especially in the American diet. As you go to any kind of fast food restaurant or almost any kind of restaurant, and the drink servings have just grown enormous. And there's a little bit of research on this. So if you think they've grown over time, in fact, you're right. I mean, for most of us in this room, you were a kid, Coke bottles were little bottles. All right, and in the 1950s, the standard size was a 6.5 ounce bottle, something like Coca-Cola. And then in the 60s, they got bigger to be 12 ounces, and then in the 90s, you got these super-sized ones. And now you have these ridiculous ones. They not only cause obesity, but it's back problems, just carrying these things around. All right. And it is a somewhat uniquely American thing, interestingly. So found this nice figure here comparing just standard drink sizes. So one of the world's standardized food options, McDonald's, you can go to one everywhere in the world, and yet what is a small and which is a large depends on which country you're in. It's not standardized. So for in the United States now, a small drink is 16 ounces, and just going back, this is where a small is now. That's ridiculous. All right. And you see that in other countries, small, medium, and large. They don't have a medium in Australia. You go to Hong Kong, they're smaller, all three sizes. You go to Japan, what's called large is only 67% of what a large is in the United States. And of course, this is really major nutritional problem for us, is that so many of the calories that constitute our daily diet come from soft drinks, and they come from very large consumptions of sugar in the soft drinks. So let's look at the chemistry of what the sugar is. So carbohydrates are made of simple sugars, and they're called carbohydrates because they're made up of carbon and water. It's hydrated carbon, basically. You take one carbon, you add one molecule of water, H2O to it, and you can get a carbohydrate. So the core sugar that's used in your body and in virtually all creatures is glucose, which has 6 carbons. And you see here you've got a carbon with H2O, carbon with a water molecule, carbon with a water. And it all works out because this one is a carbon with an oxygen, a proton, and here's the extra proton. So the ratio is 6 carbons, 6 water molecules, and you get a sugar molecule. All right. Now, in terms of the metabolism and the energy yield of this molecule, one thing that's important to remember, we're going to come into this probably not today but the next time, is that there's a lot of oxygen in this molecule. We get the energy from our food from combining it with oxygen, but much of the sugar is already combined with oxygen, and that has an important explanatory role in the relative caloric content of different kinds of food. So we're going to come back to that. All right. So there are just minor differences in the rearrangements of the carbons in the water. So these two molecules here, for example, are all identical in these bottom 4 carbons. But up here you see there's a little difference. In this one, this oxygen forms 2 bonds with a carbon in the second position. Here it forms 2 bonds with the carbon in the first position. And that gives you the difference between what a glucose molecule is and what a fructose molecule is. Same basic stoichiometry, but just little tweaks in the arrangement. Okay, and there are hundreds of different kinds of sugars. All right, so these ones that are the major ones in your body, in your food, are 6 carbon sugars. But there are other sugars in your body that play essential roles that have the same carbon in a water molecule, but are 5 carbon sugars. So this one, 2-deoxy-D-ribose, the D-ribose is what the D is in DNA. DNA is deoxyribose nucleic acid. So DNA has every base pair, every basic building block of the DNA molecule has a deoxyribose in it. All right. This one is just ribose, and that's what you use to make RNA, ribose nucleic acid.
Where do we get that?
So where we get those primarily is we make them from these. We can remove a carbon, rearrange the molecules, and get them that way. Also, if you eat DNA, which you do in your diet, because you eat cells that have nuclei, and you can chop them up into pieces, and you can recover some of these sugars from chopping up the DNA in the plants and animals that you eat.
So I'm plant-based, so I can get that from plants.
Yes, but you really can make it in your body too. You don't have to get that in diet, but there's a lot of these sugars here. And there are many other kinds too. Now, deoxyribose is called deoxy because it's actually missing an oxygen. Okay, so there's one fewer. Notice here there's an oxygen on this carbon that's not there. That's why it's deoxy. Okay, so it's a slightly altered form of the sugar. All right. Now, the major sugar in what we eat is table sugar, is this molecule right here, sucrose. And it's really a double sugar. So this is fructose down here, and that's glucose up here. The 2 sugars that I showed you in the previous slide can be linked together via an oxygen here to make a double sugar, and that's what we call a disaccharide. Saccharide is a sugar, di means you have 2 of them. This is what table sugar is. All right. And so most of the major plant producers of sugar store it for their own purposes in this type of molecule. It's very soluble, moves around in water easily, has a high energy content. So if you get maple syrup from a tree, the tree has used photosynthesis to make sucrose, and it can move it up and down in the phloem tissues in the tree, store it in the roots or below, and we can tap that and get the sugar out. All right. Now, you can easily take the disaccharide and split it to produce these 2 6-carbon sugars. And you do that simply by adding a water molecule to this oxygen here. So you see, this is H2O. If you add OH to here, all right, it ends up in this molecule, and the other proton, all right, is added down here, and it ends up in this molecule to make the stoichiometry all work. This reaction up here, that is what happens in your stomach and in your intestine. So we don't actually take up into our blood sucrose. What we take up into our blood is these two simple sugars, glucose and fructose. And it's really one of the core molecules that drives the energy production and cell building in our body. So you have the enzyme, I'm going to talk about enzymes today, that can split this, and it's not inside our body anywhere. We actually secrete it into sugar into two pieces before we take it out. Now, that's kind of interesting, important to know, because it comes up in a lot of nutritional questions about what's the effect of different sugars, and which ones are the good ones, and which ones are the bad ones. And there's a lot of misinformation, a lot of inaccuracy out there about sugars. All right, so let's go into that just a little bit. Any questions about this structure in the chemistry? Okay. All right, so here are the sort of major ways we get carbohydrates, the major sugars in our diet. So table sugar is most typically from sugar cane, and what that is sucrose. It's that disaccharide. If you eat table sugar, it goes into your stomach and intestine, the enzyme there splits it, and so when you eat cane sugar, you end up with 50% fructose and 50% glucose. This is disaccharide, you get one of each molecule for every sucrose molecule. The sugar that's in sugar beets, which is the other major source, is exactly the same. Sugar beets make sucrose, you split it, you get glucose and fructose. All right, those soft drinks that I showed in the early slides there, major source of food in our sugar in our diet, that is primarily high fructose corn sugar. That's it. Okay, and what that is composed of is it's sugar that has been converted to a mixture of fructose and glucose. Comes in several different forms for these kind of food industrial uses, but this is the most typical one. It's 55% fructose and 45% glucose. So the point here is that high fructose corn syrup is really chemically virtually identical to the standard kinds of sugar. So high fructose corn syrup is often viewed in a lot of websites, etc., as a particularly evil food product, but chemically it's just the same. You're just as bad off eating organic sugar cane as you are eating high fructose corn syrup. In fact, you think, well, what would be the best, most natural kind of sugar to get? Well, it would be honey, right? It's made by bees from flowers, etc. Well, what is it like? It is almost identical to high fructose corn syrup. All right. Honey is not made of sucrose. Honey is made of primarily fructose and glucose, and then honey has other things in it, and including probably undigested pollen, etc. So they often say you should not feed honey to babies, for example, because there is the chance that they will have an allergic reaction to some of the trace proteins that are in the honey. All right. So bottom line here, is sugar good for you or bad for you? Well, the answer is you have to have some of it. It's good for you if you have it in reasonable quantities. All right. Are the different forms of sugar, do they have different effects? Well, in terms of cane sugar versus beet sugar versus honey, all of these, they all end up producing this mixture of fructose and glucose, and that all happens before it even crosses the cells in your body. It happens in the fluid in your intestine. And so eating a grape and drinking a big bottle of some sort of soda nutritionally is very much the same thing. I kind of pick on grapes because they're almost pure sugar. All right. Now, the other way that we get sugar in our diet, one of the major ways, is by eating starch. And it is somewhat different in that starch does not produce any of the fructose sugar. It produces just glucose. Okay, so let me show you what starch is. Starch is a polymer. So polymer is the word meaning you polymerize, you string beads together on a string, and it is a polymer of glucose. So this is the 6-carbon glucose molecule linked with an oxygen to another glucose, a whole bunch of glucoses. And you can break this up into individual glucoses just by adding a water molecule right here. And as before, in the H2O, one of the H's will go to one sugar and the other OH will go to the other. And you'll end up with that ratio of 1 water for every 1 carbon. So starch is the way we get most of the glucose that it ultimately ends up in our bloodstream. So, in terms of whether one form of carbohydrate is different than another, here's a place where there is a kind of important difference. So, starch, pure glucose, all these other kinds of sugars are this mix of glucose and fructose. Alright, so does that make any difference? Well, there's a big controversy about that at the moment. So this guy, he's an endocrinologist, works with children, and he's up at UC San Francisco. So he's not a snake oil guy, all right. But he has been on a crusade talking about the hidden truth about sugar, etc. And his contention is that the real problem in our diets is not sugar per se, but fructose. All right, that the difference between fructose and other kinds of sugars does have a physiological effect. Now, to get into this really would require us to delve more deeply into the physiology of how we deal with this than we have time for, than I'm a real expert at knowing how to do. But the basic idea, and there's a lot of physiology that backs this up, is that our bodies are really designed to deal with glucose. That is what we use to circulate the main form of energy in our body. The glucose can be used to make ATP, we're going to go into. The glucose can be used to, feed our brain, etc. We regulate its level very closely with hormones like insulin. We're really designed to deal with a kind of glucose world. Fructose, on the other hand, goes to a different metabolic pathway. So it primarily goes to our liver, and a lot of glucose goes straight into lipid metabolism. What that means is fructose gets converted to fat. All right. And Lustig is arguing that that's a major problem of obesity in America and other parts of the world, is the high amount of fructose that we get in our diet. And she says it doesn't matter if it's high fructose corn syrup. If it's sucrose, it's going to produce 50% fructose also. All right. So I kind of over the years in teaching biochemistry, I've tried to do a lot of reading on this because there's a lot of skepticism from really good nutritionists. The argument is really what's the threshold, because it's not as though fructose is a poison. All the nutritionists would agree that it's important to eat fruit. So if you would eat an apple, what's in an apple? Okay, well, here's the composition of an apple. An apple is very sweet, and most of the sugar in there is fructose. Okay, so an apple has 57% fructose, 23% sucrose. When you split that, half of that is fructose, okay, and the rest is glucose. And so the consensus at the moment seems to be that yes, if you are really overdosing on sugar, then you may be exceeding some level of fructose that yet your body has trouble dealing with, or it deals with it just fine. It converts it to fat, but that has all sorts of secondary consequences. It also alters your normal kind of uptake of glucose, and perhaps that's why you lose the insulin regulation of glucose. And exactly how that happens is we still don't really understand why you get type 2 diabetes, which is a major medical problem in the U.S. So the bottom line from my reading is that as long as you're taking in these kinds of forms of sugar in your diet in moderate amounts, fructose is just fine. You're certainly encouraged to eat fruits, but there is this kind of good argument out there that one of the possible problems with excess sugar, excess sodas, etc., is the high level of fructose intake, which gets converted to fat, which has other metabolic problems. So, I don't know. Questions about any of that?
How does stevia compare?
So, stevia is one of many kinds of artificial sweeteners. All right. So, the stevia itself is like 100 or more fold sweeter than any of that. Instead of sugar, the sugar consumption can go very much down, and you're usually taking in very little of that. In other words, the actual amount of stevia or any of these other sweeteners is very tiny. And so most of the evidence suggests that these artificial sweeteners don't seem to be associated with any toxicity. But this is an interesting question of whether it actually works in terms of allowing you to take in fewer calories. So there's this one report, and I really should try to get at the original article because it's fascinating if true. Is that so? What makes something sweet? I mean, that's an interesting physiological question in itself. What is sweetness? Well, and the answer is it's a little chemical molecule. It binds to a protein that's in our tongue. It has just the right shape to be recognized as receptor, which sends an electrical signal to our brain that says, sweet, it's really good. You know, eat more of this. All right. I mean, there are other receptors in there that say, tastes awful, spit it out. You know, our mouth has evolved to do a little chemical check on everything we eat. And things like stevia, aspartame, all of that are molecules that bind more tightly, or once they bind, stay bound to these receptors to say, turn this receptor on, send this signal, and keep sending this signal. Sweet, sweet, sweet. Saying, okay, you know, you can eat more of this because we like it. It's sweet. It's sugar. It's good for us. All right. However, we have. How do you decide when you're full to stop eating? All right. You feel full after a while, right? Well, where does that come from? What's the physiology of that? Well, it turns out it's the same system. There are these receptors in your intestinal system. And they say they will also bind sugars and say, send a signal to your brain, say, got a whole lot of sugar down here we're working on. Just hang off, not sending down any more for a while, you know. They're really receptors of sugars in your intestine to say that you're full, you've had enough. And there's some evidence that these artificial sweeteners work on the ones in the mouth but not in the stomach. So they turn the on switch on, but they don't turn the off switch off. All right, and so with artificial sweeteners you can end up eating even more than you normally would, because and if there are other things that you're eating besides the sugars, then you're just eating more.
Huh.
All right. So, so it's complicated. All right.
Thank you.
Question. Yeah.
Well, when they say, in certain recipes, you use a simple syrup. So you take sugar and water, well done, and you have simple sugar. Then is that better? I mean, it works better in making ice cream, for instance, because
Yeah. I'm going to talk about it a little bit, but the answer is it's still glucose and fructose. Okay. There's almost nothing that's going to change it. That there is, interestingly, one way in which you cook your food which will make a difference. And somebody asked me this question in this class, and I hadn't heard about this. You've probably heard about resistant starch. So somebody told me about this, that if you cook a bowl of pasta and you eat that pasta, that's starch. You break it all down in glucose molecules, you take it out. What if you cook that bowl of pasta and you put it in the refrigerator? And then you bring it back out, warm it up a little bit and eat it. And somebody said, then you won't get as many calories from it. I thought, that's crazy. Well, it turns out to be right. It's right. It's right. All right. So, and here's the reason why. All right. So, getting just a little bit ahead, but it ties in. So, the other major place where we have these sugar molecules in our diet, an important role, is in cellulose. So the walls of plants are simply chains of glucose molecules. There are more complicated ones also. So piece of paper is almost pure sugar molecules, but it's a polymer in which the sugar molecules have been put together in a different order, and the key difference is they bind so tightly together that they won't bind water molecules. They won't dissolve in water. And so the one reason why if you eat a piece of paper or you eat the walls of plant cells, or like you eat a green bean, a lot of that is cell walls, you can't digest the cellulose because you can't dissolve it. The enzymes can't actually get at it. And when you do this to starch, cooking pasta, when you cool it down, it crystallizes in a form that is less soluble. And in fact, and then when you eat it, a lot of it passes through your intestine undigested. Now, not 100%. I don't know exactly what the numbers are, but I was amazed to read about this. It's a real thing. It's called resistant starch. I don't know if that works as a diet. I'm sure there are schemes out there for it. Pre-chill all your pasta. Okay.
Would that also work on rice?
It should work on anything with starch. Yes. Yes. Okay. All right. But big caveat on that one. And resistant starch is a real thing, but I don't know to what extent it actually affects caloric intake. Question over here.
I must have missed something in the chemistry in there, because we know that drinking a lot of sodas which are high fructose, or taking any high fructose, will precipitate diabetes, right? Type 2.
There's a correlation.
Okay, then why do they test for it with high glucose? The glucose tolerance test. Why isn't it high fructose?
Well, the idea is that the uptake of glucose is throwing the net balance off, and the critical effect is in diabetes is not dealing with the fructose, but the fact that it changed the regulation of the glucose levels. So they test for glucose because that's the critical level. If your glucose is being too high or too low, because your body is just responding to different loads of sugar, then that's the signal that you have a serious medical problem. In other words, the fructose test won't tell you whether you're having an imbalance in your sugar metabolism.
Because it doesn't do the other side.
Right, because you need to look at the glucose itself. I mean, one other really interesting phenomena that's just not, they can't explain at all, is there's one really good cure for people with this type 2 diabetes, and that is people who are seriously overweight will often have this bariatric surgery where they literally staple your stomach. It's found that in something like 80–90% of the cases, I mean, these people are doing it in part because they're serious diabetics. Within a week of that surgery, their glucose levels go back to normal. And it's not just food intake, because you can't mimic it with food intake. It's somehow actually changing the stomach. So it tells you that there's a real complex hormonal regulation involved here in the operation of your stomach, and if you somehow shut it down, it somehow basically cures diabetes, and they do not understand why. Okay. All right, so we'll move on here. Okay, so let me go into a few little practical things here. So, for example, if you get some French rolls at the grocery store, and you think these must be really good ones because they have no high fructose corn syrup in them. All right, but if you look over on the other side, you know, what's one of the ingredients in it? It's liquid sugar, glucose and fructose. You've just made high fructose corn syrup yourself. But by the way, why do they use so much high fructose corn syrup? It's cheap. It's cheap, exactly. It's cheap, and it also comes in a liquid form, so it's easy to use in a manufacturing process, and it's inexpensive. They make it out of starch by adding enzymes to degrade it into glucose and fructose. All right. So some other interesting things as you wander through the grocery store. This one really caught my eye because these are Heart Bar oatmeal squares, and they've got all of these good things on all these banners up here, these good things for them. And what they're telling you here is that these are heart healthy, and they put these ingredients together to create convenient snacks that are truly good for your heart. All right. That sounds pretty good. All right, well, let's flip over the other side and see what's in Heart Bars. Okay, so it's a pretty much, you know, it's a kind of a low fat, it's only 8% of fat. It's, you know, it's mostly carbohydrate. Okay, and the advertising here is that it's because it has these plant sterols. So what it has is this major ingredient is rolled oats. It's oatmeal. All right, it's oatmeal. But what else is in there? Well, the second major ingredient is date paste. Dates are very sweet. This is sucrose. It has tapioca syrup. Tapioca, that's starch. You convert that is glucose. It has invert syrup. Invert, the word invert comes from the word invertase. That's the enzyme that splits sucrose to make glucose and fructose. This is sugar. All right. It has apple juice concentrate. We saw what apple juice is. That's glucose, fructose, and sucrose. Plum puree, glucose, sucrose, and fructose. Evaporated cane juice. That's sugar. Rice syrup. That's sugar. This is an oatmeal cookie filled with sugar. All right. So, you know, it's probably a perfectly decent oatmeal cookie, but is this particularly good for your heart? No. You know, it's completely bogus. I mean, the only thing here that is maybe just slightly nutritionally valid is it says it has these plant sterols that help lower cholesterol. Well, yes, but you'll get that out of eating oatmeal in any form. Plus, if you actually go into the details of, you know, is this a valid claim? Well, I'm looking into this a little bit, and somebody did an analysis of 56 different experiments with plant sterols to see if it lowers cholesterol. Some evidence that a lot of variability, maybe lowers it 5 to 15%. And one of the interesting things is that it really seems to make a difference in what form the plant sterol comes in. So it's best if it's in a kind of combination butter or mayonnaise. This is kind of, I mean, why? That's wacky. In other words, if it's a part of some fatty thing, it's more effective than if it's a part of some sugary thing. Why is that? Because the plant sterol itself is a fatty molecule, and to have it be soluble in the food so you can take it up for your intestine kind of depends on its sort of delivery vehicle. So it may well be that all the sugar in here, the plant sterol, doesn't even ever get into your body. It just comes in one end and out the other. Anyway, and then my third one. Oh, Louise.
How about the fiber?
I'm going to come to that right now. Okay.
1 gram of sugar, wasn't it, that was in that oatmeal thing?
Well, it was, how much was it? Well, up here. So it gives you the total carbohydrates, but it's 11 grams of sugar. Yeah. All right. So here's another one I found in the grocery store. Healthy noodle. This one's got, it's sugar-free, fat-free, gluten-free, cholesterol-free, dairy-free. All right. And what really caught my attention of this one is that per serving, 25 calories. That's a really low-calorie item. So we look on the back of this one, and what's in here? Well, total fat, 0. Cholesterol, 0. Got a little sodium. Carbohydrates, nothing. You start wondering, well, what is in here anyway? There's nothing in here. 25 calories per serving.
4 ounces.
12 servings per container. This means that if you wanted to survive for one day on this, you would have to eat 100 servings of this. Well, so what are the major ingredients? Water, soybean fiber powder. This is the plant cell walls of the fiber, and cellulose. So what is this? What are these noodles made out of? Paper. You're basically, so you could eat the box. You could eat the box. You have the same effect. All right. So the objection here is not it's a bad food. It's just it's not a food at all, really. All right. But just you know, and so just in general, whenever you see all these string of claims out here and the use of the word healthy, be real skeptical, you know. So is there any advantage to something like this? Well, so Louise addresses yours. What's good in here? Fiber. Okay. It is a form of fiber. And so if you use that to put some good sauce and other good things on it, it might work out. Okay. But really useful to read the backs of these food containers. So let's look at the fiber question. Okay. So we've all heard that it's important to have fiber in your diet. So what is fiber chemically? And some fibers are kind of small molecules that are soluble, and they're important. I'm just going to focus on the long, stringy types of molecules. And so the major form of fiber in our diet is cellulose. And this is a carbohydrate because it's just a long polymer of glucose. And I said the key chemistry is that it forms a sort of tightly packed strings of glucose that won't dissolve in water. And because of that, we can't have our enzymes get at them to chew them up into the sugar molecules. And so they pretty much go through our digestive system intact. And you find this cellulose in anything that has plant cell walls in it because that forms the wall of plants, the cellulose. So cereals, fruit, vegetables. Where you do not find it is in any animal tissue because animal cells don't have walls. So they don't have cellulose, so they don't have fiber. So if you're on a really high animal, high meat diet, that's one thing you will end up lacking is fiber. Now, and these others are just other components of the walls of plants. They're all different kinds of sugar chains. I said there are hundreds of different kinds of sugars. Well, if you add other sugars besides glucose, you can get these other types of chains here. All right. things like lignin, it's really interesting. So look at those redwood trees out there. They are basically cellulose and lignin. And lignin is a really complex molecule. It is almost like a plastic, a really complex plastic, very difficult to degrade. They're just certain microorganisms that can do it. Now, why is the fiber important? All right, so this starts to stretch my physiological knowledge here. But two big reasons. One is that our whole intestinal system has a lot of bacteria and microorganisms in there. So you have this kind of fibrous, meshy network into which these organisms can attach and live. They can digest some of the fiber. Most of it goes through undigested, but they can digest some of it. And it also kind of affects the water balance inside the intestine. So you know, high fiber diets are considered good for constipation problems. And that's simply because if you don't have enough water in your digestive system, then all of those undigested materials from your food form a very hard, drier compound, and it just, your system doesn't work properly. Right? I mean, so that's a pretty superficial view of it, but fiber is really important for the microbial flora in there and for maintaining your water balance. And it's a big reason why nutritionists push eating kind of whole foods, things with cells in it, as opposed to processed food. Extracting the carbohydrates and the proteins from the cells, and they throw away the cells and just give you the inner goodies, and you throw away the fiber when you're doing that. Okay, so if we look at the various ways we can get fiber, the main point here is that if you look at meat, there is very little fibrous material in meat. Things that are really best are things that really look like kind of intact plants. So beans have a lot because often, you know, if you're eating rice or wheat, that has been shelled. The outer coating of the rice granule or the rice or the wheat seed, that's the bran, and the outer coating is gotten rid of when it's milled. But when you eat a bean, you usually eat the outer coating too, and that's where a lot of the fiber is. All right. Or in dark green vegetables, you're just eating the whole plant, so you're eating all the walls. So you kind of think of it, you know, basically, if you look at your food and you're eating something like a whole organism, then you're going to get all of the components of that organism. If you're eating a food in which you've extracted some innards from it, you're missing part of it. So you eat your potatoes, you're supposed to eat the skins. A lot of the good stuff is in there. Okay. All right. Yes?
Where do nuts come in?
So, you know, I don't really know the answer, so I shouldn't speculate. But I think nuts can be reasonably high fiber. Anybody else know? I mean, nuts is certainly considered quite good for you because the nut is a storage form of it's a seed, you know, with a lot of stored material. Nuts often have a lot of protein in it, more than grains. Okay, but I don't know how they stand in fiber. The main thing is we tend not to eat nuts in high quantities, and so often to get a lot of fiber, you just need to eat a lot of something.
We eat them with salt.
What's that?
We eat them with salt.
Yes, that too. Yeah, and they're really high calorie. So if you want to get your fiber from nuts, you just have to consume a lot of calories to do it.
I'm just Googled it. It just says fiber per 1 ounce is 2.8 grams.
2.8 grams. So that's a gram is about 28 ounces. So that's 10% fiber. So that's good. That's good. Okay. All right. So let's start looking at how we would metabolize carbohydrates. First on a big scale and then get down to the biochemical scale. So here's the major components of our digestive system. And the one thing to remember about this is all connected to the outside world. You know, there's a continuous opening from your mouth all the way through your anus. Okay, so there's a little bit of digestion that occurs up in your mouth. Then things go into your stomach. Your stomach is a really toxic environment. About pH 2, that's really acidic down there. It's just a hard place for creatures and molecules to survive. So you can kind of think of this as does the coarse grinding, coarse splitting part of the digestive system. Then it goes into your small intestine, which is so large and has such a big surface area, because this is where you're going to take your food molecules from the outside world into your cells. So you have this long tube. It's here that things like starch is being digested into glucose, and there are little carrier proteins in the walls of these cells that take the sucrose from the outside to the inside, and they can be very selective. These carrier molecules recognize the things you want to eat, and they will not take in the things you don't want to eat. Okay. And then eventually the bulk of it that's not digested ends up in your large intestine, okay, which is shown here, and comes out in feces at the other end. Now, people were asking me about probiotics, so not much related to what I do. I've done a little reading in this. I know a little bit of the microbiology. So this has become big in the last few years, and there's a lot of vagueness in the terminology. And I would say that one thing that you really have to be aware of is that when you see probiotics advertised or talked about, the way people use the words themselves is highly variable and rather poorly defined. All right. But the kind of general definition of what a probiotic is first of all, they're live microorganisms, almost always bacteria, that when administered, means eaten, in adequate amounts, confer some health benefit on the host. So that's a very general definition. But that's because probiotics are being advertised, experimented with for many different kinds of human nutritional and medical purposes. All right, so the core of it is that if you eat live bacteria, they're going to change the composition of the bacteria that inhabit your whole digestive system, particularly in your small intestine. All right, so your small intestine has a lot of bacteria. I'm going to show you some more detailed information on that in a bit. But these bacteria have a number of functions. One is they do part of the digestion of their food. So the bacteria have enzymes in them that can clip different chemical bonds, that for which you do not have any genes to encode those enzymes inside your cell. So the digestion gets done in your small intestine, and then the products are taken up in your body. Okay. And they also are kind of important because not all the bacteria you eat are good ones. All right. And the bacteria are constantly competing with each other, and if you have the good bacteria in there, they can basically kill and outcompete the bad bacteria that come in with your food, etc. So the idea is that if you had an imbalance in this population of bacteria in your intestine, you could eat live bacteria and correct the imbalance. Now there are a number of issues: which bacteria, how many, how do they survive getting through your stomach, where most of them will be chewed up as food, and get into the lower intestine. Now once they're there, it may be that they will kind of, let's say these are considered the good bacteria and these are the bad ones. Perhaps the bad ones are kind of eating some sort of nutrient in your diet, and they become very numerous, whereas this other beneficial bacteria will eat the same nutrient but remain in a smaller population. They may affect which bacteria actually stick to your cells, the cells of your body. This is rather complex, so this cartoon does not do justice to the fact that there's a layer of mucus here. You know, are you just covered with bacteria? Well, not exactly. They're sort of like a thick protective layer, and the bacteria sit on this thick protective layer. And you want good bacteria sitting there, not the bad ones. And so that's something probiotics could do. Your immune system, all right, is a way of recognizing when foreign bacteria come in. And perhaps if you put good bacteria in there, they could outcompete the bad ones. And sometimes these bacteria will actually kill each other, and the good ones might be able to kill pathogenic bacteria in them. So this is a kind of somewhat vague but overview of what's supposed to be happening with these probiotics. And so I think one of the things that's really unuseful in a lot of the literature, where take this, it has good probiotics, good for you. Well, good for what? Good for a disease, for constipation, for diet, and the answer to that is it all depends. It's such a complex system in there that you don't know what you're affecting in a good way, what you're affecting in a bad way, etc. All right, so here's kind of where the science stands now. So this is what there may be up to this number is 10 trillion cells per gram. That's a very big number. It's often been said there are 10 times more bacterial cells in your body than your own cells. Right? 10 to 1. And they are highly diverse in the kinds of bacteria, as many as 1,000 different kinds of bacteria. One reason why there's more research done in this now is that in the past it was difficult to know what was in there because you couldn't grow them in the laboratory. Some of them would grow and a lot of them would not. But now you can just take a fecal sample and get the DNA out of it and sequence all the DNA. And then you can read the DNA to see how many different kinds of organisms you have. It makes it possible to do a screen, a profile, if you will, of any individual's microbial population just by doing this kind of DNA analysis. Right now, that's still not cheap. It probably costs about $1,000, but that's doable. All right. So the bacteria make up about 60% of the dry mass of feces. It's mostly bacteria that's coming out the other end there. And it's not just bacteria. There are fungi. Protists are single-cell little organisms like paramecium. Archaea is another form of bacteria. Okay, and viruses. Just tons of these things present. And they can have an effect too. So it's really a kind of new area in science in the last 10 years. And as far as what you see in the grocery store, there's a common theme here: be skeptical. Okay. Questions about that?
So I know that I used to work writing up supplements.
I remember that. Yeah.
My undergraduate degree's in nutrition.
Yeah.
So I found out that the reason why they're considered better than foods is because they bypass the stomach, and they remain intact in the stomach, and then they continue on their way, and then they affect the other parts of the intestinal tract. Why is it therefore that yogurt or other foods containing all these products, then why are they helpful? Because wouldn't the stomach just break up the probiotics?
So the answer is that some of the probiotics can survive that low pH environment in the stomach. And if you look up what's actually used as probiotics, you know, out of these 1,000 different kinds of species, right now there are only 2 or 3 that are typically used. And one of them is much related to the kind that's in yogurt. There are some others that are kind of common in our regular intestine, and you can culture them and put them back in. But if it works, what you're doing is you're selecting for those organisms that can withstand those extreme environments. But in some cases, that's a real issue. And it also appears that you have to take a pretty high dose because there is a big loss in going through the stomach. Yeah, so there's certain kinds of lactobacillus, etc. If you look it up, there are really only 2 or 3 that are being used right now. So, you know, I think we're in the very early days of actually making use of this to address any problems. Okay. Now let's see. So we are at 10:30. How about if I spend another 10 minutes? Are we okay with that? We're at 11:30. All right. There's one more part here that I want to finish to prepare us for the kind of biochemical part I wanted to get in our third lecture. So I want to spend a little bit of time here at the end in talking about how enzymes work, because you can't understand biochemistry, you can't understand what you do with your food if you don't know what an enzyme is. And enzymes are the active components of cells. They are what does all of the work. So an enzyme is a big protein. I'm going to show you what this is. It can take a molecule like sucrose, it will bind to it, it will add the water to split that bond and produce glucose and fructose. All right. But what are these enzymes really? How do they work? So almost all enzymes are proteins, and proteins are formed by long chains of amino acids. This is an example of an amino acid down here, relatively small molecule. And typically there will be 500 to 1,000 of these in a long chain to form this big molecular structure. So amino acid has an amino group and these 2 carbons here, and this is the acid here. This proton can be released. Acid is protons, okay, or it can reattach. It can hop on and off here. They all have this same core structure, but then they will additionally have added here something else, like it can just be an H or a CH3 or another amino group or whatever, and that allows some variation. There are 20 different kinds that we need to make proteins, 20 different kinds that you have to have in your diet. Okay, so in a sort of cartoon version, so here are the 20 different kinds of amino acids have been put together in a chain, and if they're like lysine, glutamate, threonine, alanine, etc. And how they vary is some of them have no charge, they're neutral, others are positively charged, others are negatively charged, and then there are some that are kind of lipid-like, which they kind of they're insoluble in water. And whether they're negative or positive will determine whether they bind their neighbors or repel their neighbors. And so think of a bead with a whole bunch of magnets or little charged particles, some of which are minus and some of which are charged and some of which will associate with others and others which will repel. They'll come to lie together in a 3-dimensional structure depending on who binds best to what neighbor. And so when you take one of these chains and let it fold up, it'll look something like this. So this is not just a cartoon. This is a good representation of a real protein. And so you see that, like, the blues are the nitrogens, and these are the hydrogens, and the oxygens, and the carbons in the molecule. All right. And the few yellow ones, there's 2 of the amino acids that contain sulfur, and those are sulfur atoms in there. So these are monster molecules. So here's a kind of typical, this is a real enzyme, and this might be, let's say, a sugar molecule. And there will be one pocket somewhere on the surface of this protein, and it can go into that pocket. Sometimes the enzyme will change, move very slightly. This varies enormously. Some show big movements, some show very tiny movements. But it will affect the chemistry in the atoms that's surrounding this molecule that's to be reacted with, okay? Perhaps split or added to. So let me give you one sort of cartoon version of this. So here we've got this chain of amino acids with a pocket in it. And let's say you have a molecule here with 3 components, and this one has 3 components. And it will have a shape, little pockets of plus and minus charges, that exactly match what's needed to bind this red one on this part of the enzyme and the blue one over here on this part of the enzyme. And sometimes you'll get places where one of the atoms in one of the molecules here is directly connected to the other enzyme, and the other one is connected to another part of the enzyme. All right. Now, what it does here is it facilitates, it makes the reaction go so much faster, because if these were just bouncing around randomly, they would rarely hit each other at just the right angle, with just the right amount of energy to cause the right chemical reaction to go. But now we've captured them, we've positioned them right next to each other. We have other atoms changing their plus in chargeness, okay, because they're sharing or donating electrons, and that makes the reaction go faster. So what might happen in one reaction is that when they bind here, what will happen is this component will be more attracted to its neighbor over here, and it'll move one of its electrons. It no longer is bonded to its neighbor over here. So you had 3 plus 3, now you've got 4 constituents in here and 2 here. You've also lost the way that the blue one sticks to the enzyme, and so it'll go floating away. Now, when this one here gets bound to that over here, it lets go of the enzyme here. And so when it lets go, then it's no longer stuck to the enzyme, and it can go floating away. And so in this way, you ended up transferring this blue part to that red part. You had something that was 3 and 3, and now you've got something that's 2 and 4. So they're like little factories, and we have about 25,000 different kinds of enzymes. What are genes? Genes provide the instructions, put those amino acids together in the right sequence. Genes provide the instructions to make enzymes, okay? And then enzymes do all of the work. All right, so just here's the summary then. So enzymes, almost all enzymes are proteins, a tiny percent that are made out of RNA. They're formed by these long chains of amino acids. They're typically 500 to 1,000 amino acids in the chain. So they're big. They're going to have 10,000 or 100,000 atoms in them. Their function is to make chemical reactions go faster. Okay, and they all will have some sort of active site, this pocket where the reaction takes place. We have at least 10,000 in most cells. Okay, and the function of genes is to encode the sequence of amino acids. And lastly, when you're eating other organisms, you're usually eating enzymes. So they're good to eat. So what are proteins? The protein in your diet, much of that protein are enzymes. If you are eating spinach, for example, you know, something that's really green, one of the major proteins there is the enzyme, one of the most abundant proteins in all of nature, the enzyme that takes carbon dioxide from the atmosphere and converts it to sugar. But for us, it's just a source of amino acids, because we can chop those amino acids into individual pieces, take them up in our small intestine, and make new proteins with them.
What's MSG? Is that an enzyme?
MSG it stands for monosodium glutamate. Glutamate is one of the 20 different kinds of amino acids. So it's a very small part. It's just that as a, these amino acids, just like sugars, some are sweet and sweeter than others. Different amino acids have different tastes, etc. So it's just one of the 20 amino acids. They use it a lot because it has some sort of preservation properties. And it binds particularly well to some of our taste receptors. Okay. All right. So next time we're going to launch into this, but I'm going to stop there for today. So any questions about enzymes or any of that? All right. Well, then I will see you all next Saturday.